are unknowns B and appendix B.
Can it be an appendix B?
Right.
It could be, but it's not, I mean, those are just some kind of common
It
thing.
has to be an appendix A, right?
It has to be an appendix, no, C, well, your
The species.
your
It
species.
can be any species.
It could be any species,
Okay.
yes.
Okay, that would make a lot more sense.
Yeah. The only thing that's guaranteed is that your genus is guaranteed to be in C1. Okay, let's go ahead and finish up the third line of defense now and kind of put together the last piece of the puzzle. Before we do that, any questions about stuff coming up or deadlines or responsibilities or? Yeah.
The urine sample is just everything's one per group, correct?
One per group, yep. Yep, and for the urine sample, what I need is I need your flow chart, and then I need your pictures, and then what your identification was. And did everybody get the rubric? That's the first time I've ever sent out the rubric, but I don't see why I shouldn't. I mean, so you shouldn't miss. I mean, you shouldn't say, "Oh, I didn't know I had to include that." I mean, the whole grading scheme is right in front of you, right there.
For the one thing you said we didn't be able to talk pictures, like it's not higher than oil hands.
Oh, I'd forgotten I'd sent out a picture. You don't have to include it if you don't want to. I think I sent out a picture also for tributylin, didn't I? The new plates for the. I think only the positive control. I didn't send out a negative. I don't know. Okay.
So other than the videos, can you still do like bacteria?
Optional points you can do up until.
Friday?
No, I think I said day of the final, didn't I, for optional?
Oh, okay, cool.
And what did I say? I don't know if anybody's planning on going to try to get the bonus points for the video, but did I give you a due date for that?
No.
Final for that also, final day, Tuesday. End of day, everything Tuesday's end of day. I've got to get my grading done so I can get my other knee done. 15th.
November 22nd.
Are you? Okay. We are now talking about the different responses that we get to T-dependent antigens and T-independent antigens. Obviously, the fundamental difference between the two is that in the T-independent antigen situation, you don't need that involvement of the T helper cell for that B lymphocyte to be stimulated to start secreting its antibody. But there is a limitation on the T-independent response. You may know what the limitation is. It's not as full-blown of a response as you get with a T-dependent antigen. You only get a certain class of antibody produced with T-independent antigens. It's the first class of antibody that's produced in response to infection. IgM. Okay, so you get the IgM response only. You don't get that class switch over later on to IgG or IgA because you're not getting the cytokine signaling, specifically interleukin 4, that you need to stimulate that particular class switch. Why aren't you getting the cytokine signaling? Because the T helpers aren't involved. A lot of times those T-independent antigens encounter the lymphocytes that have been educated, so they have a receptor now, but are naive. They've never responded to that particular antigen. A lot of times that particular interaction takes place in the circulatory system or the spleen, which is kind of the filtration for the circulatory system. So if the antigen is present in the circulatory system, that antigen can cross-link the receptors on the surface of the B lymphocyte. It can then stimulate receptor-mediated endocytosis, and that antigen is internalized then. And that then is the appropriate signal that's needed to turn on differentiation of that cell into a plasma cell, so it can then start producing that IgM antibody. What kind of molecules typically are T-independent antigens? What kind of characteristics do those antigens typically have? Simple or complex? Simple. Simple structure, typically made out of repeating units. What are the two classes of molecules that are most commonly the T-independent antigens? Polysaccharides, lipids, okay. The proteins and nucleic acids are much more complex, and they typically are the T-dependent antigens, which give us the more robust response. So let's take a look at the T-independent antigen response. So now we've got a T helper cell involved and a dendritic cell involved along with a B lymphocyte. Do you remember which class of T helper cells, one or two, are involved in the T-dependent antigen response? Because remember that initially the naive T helper cells, which are called T helper naught, depending upon the signals they get, will develop into either T helper 1s or T helper 2s. T helper 1s typically are involved in cell-mediated immunity, and they stimulate the T cytotoxic cells to do their thing. T helper cell or T helper 2 cells are involved in humoral immunity, and they're the ones that will stimulate the B lymphocyte. So here we're talking about the involvement of T helper 2 cells. In this particular scenario, instead of a single signal to activate the B lymphocyte, we need two signals. What's one signal? Binding of the antigen. What's the second signal? Release of a cytokine by that T helper 2 cell. Those cytokines allow that important class switching to take place later on in the immune response. And so, in addition to producing that IgM, then later over we can start producing IgG or possibly IgA, depending upon the location of the antigen exposure that takes place. So here in a nutshell is the T-dependent antigen response. So we've got some T helper cells right here that are educated. They have their own T cell receptor, each one a unique receptor. Those T helper cells are presented an antigen by an antigen presenting cell, dendritic cell, macrophage cell. And this shows you some of the receptor interaction that takes place. Again, there are some receptors that we haven't talked about, CD28, CD80. We have talked about CD4. But the key factor that's important to understand here is that this antigen-presenting cell is displaying that antigen in the context of its MHC2 receptor. Because remember, T cells are MHC restricted. They only see the antigen in the context of an MHC receptor. The T cytotoxic cells are class 1 restricted. T helper cells, both 1 and 2s, are class 2 restricted. So this MHC class 2 has that antigen shoved into the little groove in that receptor, and that's what's being shown to this naive T helper cell. So this activates this particular T helper cell and stimulates it to differentiate into a T helper 2. Not a T helper 1 in this case, a T helper 2. And this particular T helper 2 starts secreting interleukin 4. Now, simultaneously, we've got these B lymphocytes also hanging around, each of which have their own receptor. And I don't know why they don't show this, but these particular cells also are binding the antigen via their receptor. So that provides the first signal for their activation. What's providing the second signal? The T helper cell now interacting with this particular B lymphocyte, producing interleukin 4. And so we've got another set of reactions taking place, this time receptor reactions between the T helper 2 cell and the B cell. So binding antigen, which isn't shown here, and then secretion of interleukin 4 once this T helper cell has bound to the B lymphocyte are the necessary signals that we need to now cause this cell to differentiate into plasma cells and memory cells. Now we've got our full-blown immune response. And under the influence of interleukin 4, we can not only produce IgM antibody, but we can get that class switched over to the other classes of antibodies as well. So this is the same diagram I showed you before. I was showing you before the T-independent antigen response that tends to happen more in the circulatory system and the spleen. And then this is showing you over here the T-dependent antigen response, which more often than not is associated with the lymphatic system, with the lymph nodes particularly. So in this particular case, again, we get the activation of the B lymphocyte by the T helper 2 cell, and that stimulates development of that particular cell into an antibody secreting cell, which can, under the influence of this interleukin 4 produced by the T helper, produce additional classes of antibody. So here's a good summary slide. This is some of the stuff that you should know to be able to answer questions on the exam. What are some of the important cell interactions that take place? And what kind of cell is activating what other kind of cell? So, what activates T helper not cells to differentiate into T helper 1 and T helper 2s? Antigen presenting cells do that. What do T helper 1 cells do? They activate T cytotoxic cells. These primarily are in response to endogenous antigens. Endogenous antigens are ones that are produced by the cell. In this particular case, a damaged cell, a cancerous cell, or a virus-infected cell. T helper 2 cells are primarily involved in the interaction with exogenous antigens. Things like bacteria and virus that are engulfed by the phagocytic cells. And when the T helper 2 cells become activated, they activate B lymphocytes to produce the antibodies. I told you I was only going to hold you responsible for 3 important cytokines in this particular set of interactions. Interleukin 2, interleukin 4, and interleukin 12. And again, interleukin 2, what's it produced by? What does it affect? Interleukin 4, same thing. Interleukin 12, same thing. And then the important interactions between receptors. In this particular case, CD receptors and MHC receptors. This particular set of information is another way of saying that T killer cells or cytotoxic cells are MHC1 restricted, and that T helper cells are MHC2 restricted. So that's just kind of stuff you're just going to have to kind of gut through, because if you don't understand those particular relationships, it's hard to keep all of this straight in your mind. Now, this is another summary slide. I'm not going to go through it right now with you. It's just another way to show you what I've shown you already. Unfortunately, there are a couple of additional receptors in here that aren't relevant. So if there are receptors shown in the diagrams that we haven't talked about, you're not responsible for them. But this again just shows you the cell-mediated immune response and the humoral immune response, and also the difference between the T-dependent and T-independent antigens. Here's a couple of other diagrams that are good summaries that don't have nearly the amount of detail in them regarding receptors or cytokines that are involved. This just more concentrates on the cellular interactions that take place between the humoral side of immunity and the cell-mediated side of immunity. And notice how the macrophages and the T helper cells are kind of at the intersection of those two particular parts of the immune response, and how they really participate in both sides of the response. But the outcome is different over here. The outcome of the humoral immunity side of the response, produce antibody. Over here, produce cytotoxic cells, which will cause apoptosis of host cells and get rid of those damaged renegade cells. This one also shows you the difference between a primary response, first exposure, and a secondary response, subsequent exposure to the same antigen. And then this slide just kind of puts everything together. You're not going to be really responsible for this part of the thing over here, because we've already tested you on first lines of defense and second line of defense. But really more over here on the third line of defense. This one breaks it down, natural immunity and artificial immunity, down into the various classes depending upon whether you have active or passive immunity. But I do think it's important kind of conceptually to understand, we've talked about this already, what the three lines of defense are doing. Barriers and entry, protection. Okay? Generalized response if the barriers are breached. And then the highly adaptive response that we get if the barriers are breached with the humoral and cell-mediated immune response. Now, there's a couple of other considerations that I'd like to just throw out there for you to think about regarding the immune system. And one very important consideration is the HIV virus. HIV virus was first identified because a subset of people started to develop certain forms of rare cancers and other diseases that you just didn't see in a population normally at that high frequency. And a lot of good investigative work was done, and ultimately it was determined that individuals in this particular infection were suffering from drastically lowered levels of immunity. And your body was not able to fight off these particular fungal infections or developments of cancer in a way that they normally was. Their immune system was depressed. And so the HIV virus could, after a long latency period, cause AIDS, acquired immune deficiency syndrome. The reason that HIV virus causes AIDS is that the AIDS virus binds specifically to T helper cells and eventually drastically lowers their numbers by killing those cells. Well, appreciating the central role that T helpers play in both sides of your immune system, it's not surprising that depression of your T helper cell population would cause dramatic decrease in your level of third line of defense immunity. Particularly, these HIV virus bind to some of those CD receptors that are so important in the interaction of T helpers with other immune cells. So when the HIV virus binds to those particular receptors, the virus then enters the cell and causes eventual cell death. And that's one of the things that they're constantly monitoring when you have AIDS, is what is your T cell count, and particularly what is your T helper cell count. So a virus obviously can have a dramatic effect on your immune system. Steroid compounds have a general depressing effect on many of your aspects of cellular immunity. And we don't really have time to get into all the details about how steroids depress immunity, but they can depress not only third line of defense, but also second line of defense, including inflammation. It can really tamp down your immune system. One type of steroid that you tend to produce when you're under stress are cortisols by your adrenal glands. How many of you have noticed over time and experienced that it's not uncommon at the end of the semester for a lot of people to get sick? Right after final exams. You make it through finals and then you fall apart. Okay? What's going on here? Probably one of the contributing factors is the amount of stress that you're under, the amount of steroids that your body is producing, and the general depressing effect on the immune system that that particular substance has. Now, medically, steroids have been used to treat inflammation and to basically mask the symptoms of injury, and that can be a real tight line to work on if you're an athlete or if you're somebody prescribing to an athlete. Because if you give them steroids to depress inflammation so that they can play, then what effect does that have on their immunity? And also, if you're medically basically counteracting inflammation, remember one of the purposes of inflammation is to limit your activity and allow you to naturally recover. And so you've got to be very careful about giving steroids to try to mask inflammation so that you can get out there and play again. You've heard many times that exercise is good for you. It is, for a number of reasons. One of the reasons that exercise is good for you is that it does tend to increase your immune system. When you exercise, your body can produce some natural compounds, including endorphins and enkephalins, which not only are natural opiates, but also are immune stimulators and immune boosters. And so if you're regularly exercising, probably your chances of the fact that your immune system will be higher will be greater. Unfortunately, as you increase in age, there tends to be a natural decline in your level of immunity. Right after you're born, your immune system is not very robust. And that's one of the reasons why getting some passive immunity from your mother through breastfeeding can be protective. Because you haven't geared up so much yet that you can start producing a very robust immune response on your own. You don't have enough immunological history. You haven't been exposed to enough antigens, for example, to really start developing a really robust immune system. But if you have some of those maternal antibodies, like IgA secreted through breastfeeding, that can help to bridge that gap. But over time, as you get a little bit older, your immune system really starts to ramp up and become robust and develop. And it continues through age about probably 30, 40 when it reaches a maximum. And then you start this gradual decline in immunity over time. And that's why elderly people are more prone to infections. There's just a natural decline in their immune system as it kind of ages and wears out, and you don't get the robust response that you once did. A very fascinating field of study has developed over the last decade that recognizes that there is a very strong relationship between the nervous system and your immune system. And that particular branch of study is neuroimmunology. You've probably all heard stories about people making these miraculous recoveries from diseases or cancers that no one can explain. Doctors gave these individuals, months or even weeks to live, and then they recovered, and they're disease-free. And a lot of stock has been played in this kind of positive attitude concept in terms of your likelihood of recovering from disease. But that was always just kind of anecdotal. But now we're finding out that there is actually a significant amount of crosstalk that takes place between the cells that populate those particular systems in your body. We used to think that the immune system and the nervous system were totally separate entities and really didn't have much interaction. But now we're finding that nerve cells can respond to cytokines produced by immune cells. And immune cells can respond to cytokines produced by nerve cells. And so neuroimmunology is attempting to study all of those particular complex interactions. And so state of mind does appear to have some effect on the robustness and the functioning of your immune system. And those interactions are probably playing a very important part in your overall ability to fight and recover from disease. So this is something you're probably going to continue to hear more and more about all the time. Okay, we've been talking about immunology in the context of disease now, and particularly in fighting disease. The last topic that we want to talk about are some applications of immunology, and particularly clinical microbiology. In clinical microbiology, we can use antibodies and their reactions with antigens in several important but distinct ways. One is we can use antigen-antibody reactions to detect the presence of disease agents. And that's obviously really important for a physician treating you to know if you're sick, what's causing that particular illness. What type of bacteria is it? What type of virus is it? Because the type of disease agent will determine the course of treatment. Another kind of wrinkle on this is that antibodies can also be measured in your body to determine what level of natural protection do you have against that particular disease. I'll give you an example in just a second, but you may or may not be aware of the fact that rubella, which is measles, is a serious disease. That's why we have a vaccination against it right now. But it is particularly serious during the first trimester of pregnancy. If a pregnant woman contracts measles during her first trimester, the effects on the fetus are usually devastating, and usually the fetus won't survive. So one of the things that doctors typically will do is to recommend to women that are of childbearing age, particularly if they are contemplating having a child, is to get an antibody measurement of your natural protection against rubella. What's your antibody titer? And there are simple blood tests you can do to determine your level of natural protection. What do you do if your rubella titer is low? You get immunized. So you can boost that particular response. So if you ever encounter the real thing of rubella, that your body can respond before your developing child is damaged. So finding out your innate level of protection is very, very useful in terms of trying to prevent disease, not just detect its presence. And then there's another application of antibody-antigen interactions that's very useful, and that's a phenomena or a process called serotyping. And that's where you use antibodies to detect disease agents, specific types of bacteria. I don't know how many of you have started to dig through Berge's manual. You don't see serotyping at all in Berge's 7th. But once you get into Berge's 8th edition, we start seeing all of this new information about different serotypes of Salmonella, for example. And these are different strains of bacteria, each of which is a little bit different from one another, which interact with a different antibody molecule. And so if we see, by mixing a culture of bacteria with particular identified antibodies, we can find out whether members of that culture belong to the antibodies related to that particular species. So it's a very convenient way to identify microorganisms through the use of antibody testing. One of the things that all of these tests have in common is the high level of specificity that's inherent in an antigen-antibody reaction. And we make an assumption. We make an assumption that if we see an interaction between these two take place, that a particular antibody is only reacting with a particular antigen, that this level of specificity exists. Another advantage that serological testing has is that it can be highly sensitive. Theoretically, with some of the serological tests that we have available right now, you can detect in a sample as little as 1 molecule of antigen if you use the proper antibody test for that antigen. So these things are highly specific, these tests are highly specific, and they're highly sensitive. And that makes them very useful in a medical setting. So let's talk first about this idea of using clinical microbiology to identify unknown microorganisms. Traditionally, these particular tests are referred to as being serology tests or serological tests. Why? Because traditionally they involve testing blood serum for the presence of particular antibodies or particular antigens. However, the scope of these tests has now been expanded, and that serological tests are now being conducted on things other than just blood serum. You can do serological testing on cerebrospinal fluid. For example, if you want to figure out if somebody has meningitis, you would be looking for particular bacteria in the cerebrospinal fluid. And you could use serological tests to do that. Serological testing can now be done on urine as well. And even saliva. So the scope of these particular tests have expanded dramatically as the technology has improved, and it's given us much greater opportunity to be able to detect the presence of either certain antibodies or certain antigens in these bodily fluids. And that can give physicians some real important information to work with in terms of diagnosis and treatment. Okay, so in clinical microbiology, we tend to have identification of microorganisms, particularly pathogens, using serological tests, falling into three basic categories. There are phenotypic tests, there are genotypic tests, and then there are immunological tests. Historically, phenotypic testing was the first type of testing that was done to try to identify unknown microorganisms. That's what we're doing right now. That's what we're relying on, is primarily phenotypic information. Later on, beginning in Bergey's 8th edition, a little bit of genetic information began to creep its way into the identification process. And that included in Bergey's 8th edition GC content. You don't see that in the 7th edition. Simultaneously, at about the same time, immunological methods started to be used to identify unknown microorganisms, and particularly using antibodies to detect particular species of bacteria. And that also was first seen in the 8th edition of Bergey's manual. So what do phenotypic methods involve? Well, microscopic morphology, macroscopic morphology, now we're referring to colonial characteristics, physiological, biochemical characteristics, detection of presence of particular enzymes or metabolic pathways, and then some chemical analysis of specific chemical composition of cell, maybe some cell wall peptides that are characterized, maybe some cell membrane lipids that are kind of unique to prokaryotes as opposed to eukaryotes. This obviously should look familiar to you. This is the whole basis of what we're doing right now to identify your unknown. We started out with microscopic information, and we went to more colonial information, and then since the last lab exam, or since the first lab exam, it was all about physiological, biochemical characteristics. So this was the traditional method that was used to identify microorganisms, and it had its limitations. It was slow, it was laborious, you needed to collect a lot of information in order to make a good identification. You're appreciating that right now with your unknown. So here's some examples of just direct microscopic examination. Phenotypic methods that we're working on and have been working on more recently involve biochemical testing. What products are formed, what substrates are being broken down. Sometimes we put various biochemical tests together into batteries like the interotube, where you can simultaneously inoculate several different media with one colony, which increases the rapidity of those tests and allows you to get a lot of information in a relatively short period of time, enhancing diagnosis. Genotypic methods are the future of bacterial identification. You don't have to culture things. They're very precise, give you quick results, and are oftentimes highly automated. So DNA analysis through hybridization, PCR. And really what's in the game right now in terms of available techniques is not just identifying little snippets of the genome of a microorganism and trying to identify it from this limited information, but from actually doing whole genome sequencing. Genomic sequencing is becoming really, really cheap. And it's probably not going to be too long before all this other stuff is going to go by the wayside, and there will be automated machines to take a sample from you, and that they will just sequence the DNA of whatever bug is in the sample and immediately give you a very precise answer about what that is. Immunological methods are still very useful. and will still have a high degree of usefulness, particularly in more the realm of detecting whether you have a particular disease as opposed to the organism, but also determining your level of natural protection against that particular disease, your antibody titer. Again, immunological methods have to be both specific and sensitive, and they are. One of the things that's important in all of these serological methods is that you have to be able to tell or detect whether a particular antigen and antibody reaction has taken place. If I mix together two things and I want to see if there is an antibody in one sample, so I mix a particular antigen with that, I need to know if they're interacting or not. It's most useful if that interaction between antigen and antibody is visible. If it gives you something you can see easily, like an agglutination reaction, which is just a clumping. You can actually see a clumping form on a slide, for example, when you do human blood testing, which involves antibody-antigen interactions. Or maybe you get a color change that you easily can determine. But sometimes the antigen-antibody reactions aren't visible to the naked eye. So then you've got to kind of get creative to determine, I can't see this reaction occur directly, but is there some indirect way that I can measure whether it took place or not? And there's some really creative approaches that we'll talk about that have been able to bridge that gap of being able to detect antigen-antibody interactions taking place when they're not easily visible to the naked eye. So this particular diagram shows over here on this side the selectivity that antigen-antibody reactions exhibit. So here we've got two particular antibodies, and they react only with two particular antigens in this mixed antigen population. So there's a high degree of selectivity. And this particular antibody should not interact with this antigen or this antigen. It should only interact with its partner. The same goes for this green antibody. So selectivity is really important. This shows the phenomena of sensitivity. Okay, here we've got a whole bunch of antigen molecules and a few of these. Antibodies can interact with these very few antigens because of their specificity, and we can detect the presence of these particular few antigens because of this antigen-antibody reaction that took place. So literally, we can find the needle in the haystack by using this antigen-antibody specificity. So this illustrates kind of the two general categories of serological tests that I've just talked about. The one on the left involves analyzing patient serum for the presence of particular antibodies. How do we do that? We mix particular antigens with that patient's serum that would specifically interact with the antibody we're looking for. So we're doing a test, for example, for the rubella antibody. What's your level of protection? Do you need a shot? How do we do that? We take purified rubella antigens and mix them with the blood serum. If the antibodies are present, they'll react with the antigens in a very specific way, and hopefully will give us some type of visible macroscopic reaction. Some way to detect whether that reaction took place. Some way to determine whether there were antibodies for that antigen in that patient's serum. Over here on the right, we have the serotyping example. So here we've got a colony of bacteria. We want to know what it is. We might suspect that it is a particular strain of salmonella. So if we have particular antibodies that react with that strain of Salmonella, we can take some of the material from that colony, mix it up in a saline solution, for example, just to get it in solution, and then add those particular Salmonella antibodies. If this is Salmonella, it'll react with the antibodies, and we should form some kind of visible, detectable reaction. So a very precise and fast way to identify that particular species. But the key is to be able to have a detectable complex that forms. So that's where we'll pick up next time, is we'll talk about different ways you can detect antigen-antibody complexes that form during serological reactions. I have posted this last set of slides and lecture notes and things, so those are on Moodle.